In this video, we will talk about meiosis and mitosis.
First, we will be talking about the steps of meiosis.
The first step of meiosis is interphase. During interphase, the chromosomes duplicate themselves.
The second step of meiosis is prophase. It is the longest phase of meiosis. It takes up about 90% of cell division. During this step, chromosomes coil up and become shorter. Then in a process called synapsis, homologous chromosomes made up of two sister chromatids come together as a pair, which is known as a tetrad.
The third step of meiosis is metaphase. During metaphase, chromosome tetrads are aligned on the metaphase plate. Each chromosome is condensed and thick, with its sister chromatid attached.
The fourth step of meiosis is anaphase. During anaphase, it starts with chromosomes moving towards the two poles. Sister chromosomes remain attached, and only the tetrads split up. Because of this, you still see doubled chromosomes moving toward each spindle pole.
The fifth step of meiosis is telophase. During telophase, chromosomes arrive at the poles of the cell. Each chromosome consists of two chromatids. Then cytokinesis occurs and two haploid daughter cells are formed.
The sixth step to meiosis is prophase 2. During prophase 2, a spindle forms and it moves the chromosome toward the middle of the cell.
The seventh step of meiosis is metaphase 2. During metaphase 2, chromosomes are aligned on the metaphase plate.
The eighth step of meiosis is anaphase 2. During anaphase 2, centromeres of sister chromatids finally separate. A chromatid from each pair moves towards opposite poles of the cell.
The ninth step of meiosis is telophase 2. During telophase 2, nuclei form at the cell poles and cytokinesis occurs at the same time, ending meiosis with 4 daughter cells.
Next, we will be talking about mitosis.
The first step of mitosis is interphase. During interphase is a period of cell growth when the cell synthesizes new molecules and organelles. Prophase is the first step of mitosis. During prophase, changes occur in both the nucleus and the cytoplasm. In the nucleus, chromatin fibers become tightly coiled and folded to form chromosomes. In the cytoplasm, the mitotic spindle begins to form microtubules that grow out from the centrosomes that begin to move away from each other.
The second step of mitosis is metaphase. During metaphase, the mitotic spindle is fully formed with its poles at the opposite ends of the cell. The chromosomes convene on the metaphase plate. The centromeres of all the chromosomes are lined up on the metaphase plate.
Anaphase is the third step of mitosis. During anaphase is when two centromeres of each chromosome come apart, separating the two chromatids. Once they are separated, each sister chromatid is considered a full-fledged chromosome.
The fourth step of mitosis is telophase. During telophase, cell elongation that started in anaphase continues. The daughter nuclei appear at the two poles of the cell as nuclear envelopes that form around the chromosomes. Also, the chromatin fiber of each chromosome uncoils and the nucleoli reappear.